So our first student is McKenna. And what I will always remember, McKenna, is that when you apply, you have to write an essay, basically explaining why you want to be here at Max Planck during the summer. And she really wrote one of the best essays I ever saw in the 5 years I am at Max Planck. I literally sent it around to our communication, like, look at this. And found out, of course, that she's the editor of her school award-winning magazine. So it makes sense that she's so good with communication. And actually, one of her goals is to be able to explain science in a way that is accessible to everyone. And I really think she's doing a great job. So McKenna, if you want to start.
Hello everyone, my name is McKenna Senzin, and this summer I was lucky enough to be an intern at the Bolton Lab, and today I get to share with you my exploration on neural circuitry in autism spectrum disorder models. So first, I just want to start by going over autism spectrum disorder. So worldwide, there's over 75 million people diagnosed with this disorder, and it looks very different for every individual. And what makes it really complicated in terms of research is that there's no single gene that leads to the progression of the disorder. And if you look at the graphic on the left, you can see that there's many different symptoms associated with ASD. And those range for every individual to not having them at all or to having them at increased severities. So there's many different ways to go about studying ASD. But in the Bolton Lab, we look at it specifically through neural circuitry. So here on the left, you have a picture of just neurons in the brain, what you would think of. And you can see that it's pretty tangled and it's pretty hard to tell what exactly is connected to what. Think of it like a tangle of necklaces you have in a drawer. And if you look at this clump of tangled necklaces, it's really hard to decipher which charm is related to which gene. And if you just start picking at it, you're gonna break it, and no one wants that, especially in neuroscience. So instead, local circuitry takes the approach of looking at each knot and following exactly where it leads. And by doing that, you can find the entire connection within this clump of neurons and make it something into this, which is a video, and you can see each neuron light up as it's being activated. In my project specifically, I looked at the amygdala, which is known as a key region involved in ASD symptoms. Specifically, the amygdala is an almond-shaped region that regulates social and emotional behaviors. And I looked specifically at the central amygdala, which is dense with a cell type called inhibitory cells, which just stop the communication of other cells. And here is an image that I took showing the central amygdala. And you can see that there's this red cluster of cells in the middle, right here. And this red cluster is somatostatin expressing neurons, which is a subclass of inhibitory cells. And so this is where I'll be focusing most of my project on. And in order to study ASD, we need to create a mouse model that lets us look at those specific symptoms. So in my lab, we've been looking at the P10 mutation. P10, or phosphate-intensive homolog on chromosome 10, is known as a potential candidate gene for ASD, and it's often involved in research with cancer. So mice had a conditional knockout of P10 in somatostatin-expressing neurons, and that just means that the mutation was only caused in those specific cell groups, which, remember, was the red group that I showed you prior. And my lab did a bunch of behavioral testing to ensure that this mutation caused a good model for autism. So one of those tests was known as the elevated maze. And here you can see that there's these open arms that you can see the outside and these closed arms. And how it works is that mice are naturally very curious creatures, and they want to go explore their surroundings similar to how humans do. But if a mice has elevated levels of anxiety, they might get afraid when they're out in this open area because they can see visually the drop below them. And even though it's the same drop in the closed arms, it feels safer because they're not getting that visual stimulus. So if we look at the data from that experiment, you can see that the genetically modified mice in blue, known as flocks, stayed in the closed arm, the more comfortable space, for a longer period of time. And another behavioral test that was done is similar, but it's known as the light-dark box. And this, instead of having an open arm that is the novel environment, you have this open box that has a lot of bright light stimulus for the mice. And here in this data, you can see that the mice stayed in the light zone that were genetically modified for a less amount of time, meaning that they found more comfort in the darker area, even though they're naturally curious creatures. So With a combination of these experiments, as well as several other behavioral methods, it was found that the PTEN mutation caused those elevated states of anxiety and fear. So now that we know the behavioral effects of this mutation, my job was to quantify the effects of the genetic mutation within the central amygdala. So I wanted to observe any changes that I found. And how I did that is I imaged across genotypes. So I took mice from both the control and genetically modified, and I sliced their brains on the vibratomes to make the tissue thinner so I'd be able to image them using microscopy without damaging any of the neurons. And then I stained them so I can isolate different cell populations and mounted them so that I was able to image them on a 980 confocal microscope that lets me see the different fluorescence. So here's an example of an image that I took. Here we can see this bright region is again the central amygdala, and if we zoom into the center, taking another image with a higher objective, you can see those isolated cell groups. So those red groups, remember, are the somatostatin neurons, which are the inhibitory cells, where the blue cells are known with DAPI staining, which is going to highlight the nucleus of every cell in the region. And from there, I wanted to do two things. I wanted to count the relative number of each of those cell types in the central amygdala, and I also wanted to look at the average size of the somatostatin expressing neurons. So how I did that is I separated this color channel so I can isolate those cell populations, and using a system I developed on ImageJ through Fiji, I was able to get that data. And I did this entire process blind, so I did not know the genotypes of the mice, so I can avoid bias when doing my counting. And just to put some context on this, I had 10 brains, as I said earlier, and the central amygdala is about 800 microns thick. And since my slices had to be 100 microns thick so I can visualize them, and that the amygdala is in both hemispheres of the brain, that's approximately 300 images that I took, and I counted about 36,000 cells. And from that data, this is what I got. So this is showing the ratio of the somatostatin expressing neurons to all other cell types in the region. And we can see that the genetically modified group in blue is having a higher ratio. So it just means that there's more somatostatin cells than compared to the wild type. And with the average size, we found something similar where that the genetically modified group had larger cell sizes on average. And after running through a series of statistical analysis, the data on the right with the average size was proven to be significant, where on the left, it was close with a p-value of 0.06, but it's likely going to be needed a bigger sample size to ensure that this trend is true amongst all individuals. And from there, what I wanted to do was trace the dendritic arbors to observe structural changes. So dendritic arbors of a cell are essentially the branches that come off of the neuron that let the neuron receive signals from across the region. And the reason that I was tasked with doing this was so I can test if a free software would get comparable results to the data previously acquired in my lab doing something very similar. And so the process for that starts with my mentor doing an experiment known as circuit mapping to find cell communication. So this is done in an ex vivo state, meaning that the cells are acting as if they are in a live brain for approximately 6 to 8 hours. And in that time, you can see what would be going on if you were actually activating these in a live animal. And so what you can do is you patch onto a specific cell, and that will allow you to read the electrical signals given off from that cell. And then you can pinpoint other cells in the region and non-invasively stimulate them using a laser one at a time. And so by choosing specific time increments to stimulate them each and looking at those electrical signals, you can see which cells are connected to the one that you're patched onto. And you also are able to take a calcium video similar to that video that I initially showed you with the neurons lighting up to ensure that the stimulation with the laser is working. So just to summarize, if one laser beam is pointed at this cell and you get an electrical signal to a certain threshold over repeated experiments, you can make the connection that there is a connection between the cells. And from there, you can fill the cell that you're patched onto with biocytin. And biocytin is a chemical that is going to seep into the dendritic arbors of a cell. So something to look like that, so it's easier to visualize. And from there, this is the image that we acquired. And it's pretty dense, just like most areas of the brain with neurons, but the one that actually has the biocytin fill is the one right here in the middle. It's a little easier to see if we separate it into color channels. So if you guys remember the DAPI staining, which highlights the nucleus of all cells in the region, you can see that present here, but you see how the cell in the center is not a circle unlike the rest of them. And you can also see these faint lines coming off of it, which are the dendrites. And so by going through different layers of Z, I was able to trace these very faint lines and follow them throughout the region of interest. And from there, I was able to create this 3D construction of the neuron in the cell. So you can see here there's the center of the cell going around, and those are all of its arms branching out in the region. And from that data, I was able to glean this graph. So this is showing the dendritic intersections of wild-type mouse. And I, again, did not know that the image I traced was wild-type until very recently, so I could avoid bias. But we can see here that the data is shifted towards the left of the graph. And that just means that the majority of the intersections are towards the center of the cell. And if we compare that to the data previously acquired in my lab, you can see that the black lines match up nicely where they're both shifted more to the left with a peak in a relatively similar location. When you compare that to the genetically modified group, you can see how they have a lot more dendritic intersections going further out across the region. And again, that just means that their dendrites, or their arms, are being stretched out further. And so from this comparison, it was clear that the free software can get comparable results to the more expensive one that my lab previously used, and it might be used moving forward to save money that could be allocated to other research tasks. So in conclusion, in my 6 weeks here, I was able to observe the PTEN mutation in mice that created an elevated levels of anxiety and fear in mice. And I found that the mutation caused an overgrowth in somatocyte in quantity, size, and dendritic arbor lengths. So this research will be used in context in the next chapter of this neuroscience research, which will aim to find common circuitry paths across this model of ASD and others. And so hopefully by observing these structural differences and by finding those common circuitry, it can become much more clear what exactly is going on in the central amygdala that leads to these symptoms of autism spectrum disorder. So I wanted to, of course, thank all the people who were able to get me to this point. I want to thank the Bolton Lab and Dr. Bolton for letting me into her lab space and acting on my curiosity. I want to thank my mentor, Tim, who taught me all of this, and that's pretty impressive, and also helped me be able to communicate with all of you. I wanted to thank Caitlin, who was a lab technician in our lab, who helped me get all those amazing images you were able to see. And of course, I want to thank MPFI Education for allowing me this opportunity. Thank you, Erica and Ilaria, for believing in my essay and making me be here today in front of you. And then, of course, I have to thank the MPFI supporters and donors who allowed me this opportunity and give students across the county experience in science. And so now I would gladly answer any questions any of you have.
Do we have any questions? It's early in the morning, so usually.
So the counting is done. Oh. So the question was, how is the counting procedure done? And so depending on the results that you're looking for, there's many different ways to go about it. And so what I did is I went through dozens of science blogs with different scientists comparing, oh, this is my favorite way to cell count. No, this is the best way. And based on all the pros and cons that I looked at different softwares, and I found this plugin on a free software, ImageJ, that I mentioned, and I created a pipeline essentially that allowed me to count it. So first, what I was able to do was I thresholded the image, and that just pinpointed the brightest points in the image, which typically were the cells that I was looking for. And if any cells were cut out of that, I would add that to the counting later. And from there, I did a couple image adjustments so that you can divide the cells that were overlapping. And for the DAPI, because it makes 2 dots in every cell, it sort of combines that into one. And then I was able to count the particles left in the image. And that gave me the data both on the number and the area, which is what I was looking for. Sure.
Any other question?
Showed a p-value 0.06, and you were not too happy with it. You wanted, I guess, a p of 0.05.
Yes, I mean, that is the number that you can used to make data statistically significant. I wasn't necessarily disappointed that it was 0.06, but it just gave a sign that more data will need to be acquired from that.
Thank you so much.
Yeah.
Great job.